Hello my dear students and welcome back to Excellence Batch, and I am your Diksha ma'am. So here we have started the chapter Biotechnology Principle and its Processes. So we have continued with the tools of recombinant biotechnology, and in today's class we'll be doing the same. So in the tools of biotechnology, recombinant DNA technology, we had one tool, a very important tool, that was vector. Sometimes they are also known as cloning vectors. Sometimes they're also known as cloning vectors. What are cloning vectors? These are basically vehicles. What are they? These are basically vehicles on which your foreign DNA, it sits, and then they move and they throw your foreign DNA or take your foreign DNA along with them into the host. So this is the main function of vector. So vector is something that we need so that it can carry the foreign DNA into the host organism, right? So what's a cloning vector? These are vehicles or carriers which carries foreign DNA into host organism, right? So there are different kinds of vectors. There are plasmid, there are viruses, right? There are different bacteria. So various types of vectors are present according to the host organism and what kind of DNA you want that to get replicated. Fine. But one thing that every vector should have are certain features. So let's see, what are these essential features? So there are certain essential features that your vector should have. Now what are these features? The first feature is ORI. ORI is origin of replication. If you have studied the DNA replication in your botany section or in the chapter molecular basis of inheritance, then you must have studied one sequence of DNA, that is origin of replication. So these are some sequence of nucleotide that are very much important if you want to start a DNA replication. For example, if I have this piece of DNA, and in this DNA, this is origin of replication. These are some sequence of nucleotide. These are origin of replication, and this is any gene. So if this gene needs to replicate, it should definitely start with the origin of replication, or basically in short I will say these are some sequence of DNA from where the replication starts. That's why they are known as origin of replication. Okay, so what is origin of replication? These are sequence of DNA from which replication start. And you know in gene cloning, this is the only thing we are doing, making copies, doing replication. If you don't have origin of replication, then replication will never occur. So that's why if you are using any vector, that vector definitely should have origin of replication, because that's a point from where replication will start. If there is no origin of replication, make sure you join it with your foreign DNA and then send the foreign DNA into the vector. But origin of replication is important. Without this ORI, replication will not occur. So without ORI, replication will not occur. Another important point to note here is that this ORI is different in prokaryote and different in eukaryote. If you have to replicate eukaryotic gene, make sure your plasmid have origin of replication of a eukaryotic gene. If you want that a prokaryotic gene should replicate, or your foreign DNA is a prokaryotic gene, then make sure the origin of replication should be a prokaryotic. So depending upon the gene, ORI is added. Depending upon the gene, the ORI is used. or added, because these vectors mostly they are modified. You make it, you engineer it. So if you know you want to use this vector for the replication of a eukaryotic gene, make sure it should have the origin of replication of a eukaryotic gene, right? So what type of gene we are adding? It can be a prokaryotic or eukaryotic. So if you are having a prokaryotic origin of replication and the gene is a human gene, it will never multiply. Okay, if it will multiply, other some sort of problems can occur later on in replication. So make sure origin of replication should be a eukaryotic. Anyways, let's talk about the second feature. The second is the most important feature, and that's selectable marker. What is a selectable marker? Selectable marker is a particular gene present in your vector that will help in the selection of different types of host. Now, what are these different types of host? Let me explain you that first, then we'll go to the selectable marker. Now, there can be a chance that you have this host organism, and this host organism does not have vector or plasmid, or during transformation accidentally the plasmid or the vector did not enter inside this. So this type of a host organism is a non-transformant, host. So non-transformant host is a host in which your vector accidentally is not able to enter. So first chance can occur this. Whenever you are doing gene cloning, when you have made the recombinant DNA and you are asking that DNA to enter inside your host organism. So there can be different type of scenarios and chances. That first chances, maybe your vector is not able to incorporate inside the host. Second reason can be, maybe your host get the vector, but this vector does not have a recombinant DNA. So this type of cell is a transformed cell, is a transformant but non-recombinant. Why non-recombinant? Because it does not have a foreign DNA. It's just the same vector. Accidentally, maybe it was not able to get cut by restriction enzyme and the foreign DNA didn't get incorporated into it. So it's the transformant because it gets the vector, but non-recombinant because it does not have the foreign DNA. So transformant because it got the vector, non-recombinant because it does not have a gene. Now another scenario can be this, where the host organism have got the vector and it does not get any, it has got the vector and it also has got the foreign DNA. So such case will be transformant because it has got the vector, recombinant host. So three kind of scenarios are formed here. In that group of bacteria which are like unicellular organisms, and you don't know, you cannot see that with the naked eye, you don't know which one get which gene. You have to select that which is my useful bacteria. Out of all these, which is your useful one? This is your useful one. So how will you identify which bacteria has got the vector, which has got the right vector or not? So to find that out, what we add some genes known as selectable markers inside your vectors to make sure whether your gene, the foreign gene, has been added into the vector or not. So basically they will confirm which is transformant or non-transformant and recombinant and non-recombinant. Okay, so what are these? These are genes to confirm the transformant or non-transformant, transformant and non-transformant, and recombinant and non-recombinant. Now what kind of genes are these? These are very simple genes. These can be the genes which are antibiotic resistant genes. Antibiotic resistant gene. And there is one more gene, that is beta galactosidase, producing LAC-Z gene. So LAC-Z gene you must have done in the lac operon model, where beta galactosidase is secreted by this gene. Now, if I talk about antibiotic resistant gene, there are different types of antibiotic resistant gene. Now, what are these? Let's see. Antibiotic resistant gene. Like we have antibiotic resistant gene for ampicillin, tetracycline, chloramphenicol, and kanamycin. Now, what are antibiotic resistant gene? We also have discussed this in the previous class. These are certain gene, which confirm that if that your bacteria is resistant to an antibiotic, or makes a bacteria resistant to an antibiotic. For example, if a plasmid have ampicillin resistant gene, then that plasmid, if it is in a bacteria, the bacteria can easily survive in the presence of ampicillin, or this gene is making the bacteria resistant towards the ampicillin. Got it? So these are the various selectable markers. The a third feature that a vector should definitely have are the cloning sites. What are cloning sites? So for example, you have a piece of DNA and that is AAAA, GAATTC, AAA. TTTT, CTT, AAG, TTT. So that's a piece of DNA. Okay. If I want that my foreign DNA should be placed inside this piece of DNA, then it definitely should have a restriction site for a restriction enzyme. For example, it has a restriction site for, as you can see, EcoR1. So that's a restriction site for EcoR1. So your vector should definitely have restriction sites for various restriction enzyme. By that you can easily cut the piece of DNA and put your foreign DNA inside of that. Right? Now, one thing that you should keep in mind, that you can have number of cloning site for different types of restriction enzyme. For example, if I have a vector, I can have the restriction site for EcoR1, BAMH1, SAL1, PSD1 or HIN2 for everyone. But for one restriction enzyme, I should have only one restriction site. For example, if I have this piece of DNA, imagine this is a piece of DNA I have. If it has restriction site for PSD1, PVU1, BAMH1, EcoR1, then it's a good one. I have a lot of different types of restriction enzyme. Now I can put any or number of kind of foreign DNA, because you never know, your foreign DNA might be get cut with EcoR1. It might not get cut with BAMH1. So we need to put different types of restriction site of different types of restriction enzyme. But we never do this thing. What is that thing? We will never put the number of restriction site for the same restriction enzyme. Why? For example, you want that your foreign DNA should go into this piece of DNA, this one. Okay, but accidentally it went to this one. Then you will not be able to identify where your piece of DNA has gone, because you will be using EcoR1, then it will make two cuts. But if there is one EcoR1 site, you will get to know, yes, my piece of DNA will enter into this gene only. So it is said that we can have number of restriction site for various restriction enzyme. The vector do not contain number of restriction site for same restriction enzyme, right? So this is good one. and this is bad one. This should not occur. Otherwise your gene cloning will be very difficult in this kind of a case, but this kind of a case will be more helpful because now you have an opportunity to use various number of restriction enzyme, right? So that's about the three main features, and fourth feature is size. The vector should have an appropriate size. If it is too large, very large, then the chances of breakage and shearing is more. Shearing means to get break, and maybe it will not be able to enter into the host. But if it is of average size, smaller size, but it has a lot of space to incorporate the large foreign DNA, then it will be the good one. So the size should be small. Should be small, but with large insert range. What is insert range? For example, if I have this plasmid, it should have the maximum size to incorporate the foreign DNA. That is the insert range. If it has a very little space to insert for the foreign DNA, then it will not be the good one. So size is also one of the very important factor when you are talking about vectors. All right. So the vector that we are going to discuss first is your favorite plasmid. All right. So now what is this plasmid? So you must have learned about plasmids in bacteria. So plasmids are circular. As you can see in the diagram, this is one plasmid. Plasmids are circular, extrachromosomal. Extrachromosomal means they are not the part of the main chromosome, they are present outside to it. Double stranded, they have double stranded DNA, not the single stranded DNA, and they are self-replicating molecules. And they naturally also have antibiotic resistant genes. So it's a good thing, it's a good deal for us. If they have antibiotic resistant gene, so they are a kind of selectable markers, and they can help us in identifying the transformants and non-transformants. Apart from that, these are self-replicating, they can replicate their own, so it's also good for us. Why? Because if you'll see this one plasmid, it has origin of replication, ORI, origin of replication. Fine. Otherwise, where can you find them? You can find them in most of the bacteria and yeast. Most of the bacteria and in some yeast, you can find these plasmid. Now, this plasmid is PBr322. PBr322. So, you must be thinking it has a very weird name. PBr322. P stands for plasmid. BR is from the scientist who engineered them, that is Bolivar and Rodriguez, and this is the number, index number, just like a roll number that you get in your class, by which you can differentiate, you can be differentiated from the other student. So if you can see here, what are the various genes we have in PBr322? The first gene that we have is ORI, origin of replication. And that is very important because from here the replication will start and then your gene can easily multiply. Then you have this ROP. ROP will produce some proteins. You can see here ROP. What is ROP? ROP will form proteins which is required for replication of plasmid. Right? So this is a gene. Now you can see on ROP you can find one restriction site for PVU2. Next, we have two selectable markers here. One is ampicillin resistant gene, another is tetracycline resistant gene. The foreign DNA is always added in the selectable markers. Mark my words and please write this down here in the selectable markers also, that foreign DNA is always added in selectable markers. In selectable markers. Because if you will not add in the selectable marker, then you will never get to know about the transformants and non-transformants. All right, okay. Moving to the next. So we have an ampicillin resistant gene and a tetracycline resistant gene as selectable markers. The size of PBR322 is 4.3 kb. Kb means not kilobytes, kilo base pairs. 4.3 kb is its size. Ampicillin resistant gene have the restriction site for PST1, PVU1, whereas tetracycline have the restriction site for BAMH1 and SAL1. You have to draw this structure around 5 to 10 times until or unless you remember it nicely, because directly they will ask you question from which tetracycline or ampicillin which have which restriction site. Okay, then the one which are present outside to it are ECO R1, CLA1 and HIND3. So these are the all restriction sites that are present in the PBR322. But the most important that you need to remember is that PST1, PVU1, PP11. Remember by this. PP11. They are in ampicillin, right? Because ampicillin have P. Tetracycline have BAMH1 and SAL1. Okay. Now, how are we able to select the transformants and non-transformant? So there is a concept behind this, that how adding a gene in antibiotic resistant gene help us to identify between transformants and non-transformant, and that concept is insertional inactivation. Insertional inactivation. Insertional inactivation. Right, so what happened in insertional inactivation? Insertional, that means we are inserting something. Inactivation, something is getting inactivated. So when we add our foreign DNA in a selectable marker, imagine this is a selectable marker, tetracycline resistant gene, and we used BAMH1. We have used BAMH1. By using BAMH1, we have added our foreign DNA here. What have we added? Our foreign DNA here. So when we will add another piece of DNA in a selectable marker gene, the selectable marker gene will lose its identity and it will not be able to work. Imagine there is a gene which is tetracycline resistant gene. It's doing its work. It is making the bacteria resistant to tetracycline. If you will add another piece of DNA in it, then tetracycline resistant gene will get destroyed. It will get inactivated. Now when it is getting inactivated? When you are inserting a foreign DNA. So this concept got its name insertional inactivation. It got its name insertional inactivation. That means inactivation of a gene, inactivation of selectable marker gene on addition of foreign DNA. On addition of foreign DNA. Right. So this will help us to know that your bacteria is carrying the plasmid or not. Okay. So let's elaborate this one. So how do we do this all bacteria growth and all? How do we do bacterial culture and how do we grow these colonies? Let's see. Imagine this is a plate where your bacterial colonies are growing. Now you need to identify out of these which are transformants and which are non-transformants, and which are recombinants and which are non-recombinants. Okay, so we have one transformant, one non-transformant. So we have transformant bacteria and then we have non-transformant bacteria, right? In transformant we have two categories. One is transformant recombinant and one is transformant non-recombinant bacteria, right? So NRT means non-recombinant transformants. That means it's type of bacteria which have the plasmid, the plasmid does not contain any recombinant gene or it does not contain a foreign DNA. And next we have is the another bacteria, the bacteria which have got the plasmid, and the plasmid also have the foreign DNA which is the desirable one. This is the one which we need. And the last one is the non-transformant, which does not contain any kind of plasmid. Alright, so these three scenarios are there. We have to find out which out of these colonies, these are bacterial colonies, right? Out of these colonies, we have to identify which colony of yours is transformant, which is non-transformant, which is recombinant, which is non-recombinant. Alright, now let's see. So first of all, what we will do, we are assuming that we have used BAM-H1 as restriction site. So what restriction enzyme we are using? BAM-H1. If I am using PBr322 as plasmid or as vector, what are we using? We are using PBr322 as our vector, and we are using BAM-H1 as our restriction enzyme. So it's quite obvious that our foreign DNA will go inside this gene, which is tetracycline resistant gene. Okay, so the selectable marker in which your gene is going, the foreign DNA is going, is tetracycline resistant gene. Clear? Right? It's quite obvious. So if you are using PBr322 and BAM-H1, then which selectable marker will get the foreign DNA? Tetracycline resistant gene. So first of all, we have to find out which bacteria got the plasmid and which bacteria do not got the plasmid. That means the first point of identification is we have to identify which one is non-transformant and which one is transformant. Okay, and how can you identify transformant from a non-transformant? Just by selecting the presence of the plasmid or not. So the first antibiotic that we will add in this culture is ampicillin. So what we will do, how we will do it? We will take a replica of this. The plate which we have taken is a replica of the original plate, right? So how the replica is taken? Just like we put stamp on a paper. We will take a wooden block, we will wrap a velvet cloth on it. For example, this is the original plate with bacterial gene. We will put that stamp on that. Now on the stamp you will get the bacterial colonies, and now on a fresh plate you will put the stamp, right? So how did we take the replica? First, from this original culture we will do cut copy paste, right? What we will do? We will do cut copy paste. How we cut copy paste? With the help of the piece of wooden block and velvet sheath. So for example, this is wooden block, and on the wooden block we have this velvet sheet wrapped on it like this. So first we will take a stamp, put it here, and then put it on the fresh piece of plate. Now you have got this bacterial colony. Now what we will do? We will add ampicillin antibiotic. So any bacteria, if it has plasmid PBr322, then it will be having ampicillin resistant gene. You got it? If I say this bacteria have plasmid, this have plasmid, so they both will be having here ampicillin resistant gene, because this plasmid is PBr322 and PBr322 have ampicillin resistant gene. Got it? So now the bacteria which have the ampicillin resistant gene, that means that bacteria have the plasmid. Okay, so the bacteria which have the plasmid have the ampicillin resistant gene. So you can see when you will add the ampicillin, some of the bacterial colonies, they will die. Now the bacterial colonies that will die, which are those colonies? Those colonies which die are the one which are non-transformant, because they are not able to survive under the ampicillin presence. That means they don't have ampicillin resistant gene. So these bacterial colonies, they will die in the presence of ampicillin antibiotic, because the non-transformant don't have the plasmid and they don't have the ampicillin resistant gene. Okay, so why did they die? There is no plasmid, no ampicillin resistant gene. Okay, so these one they die, these one they survive, they die because they are non-transformant, they don't have plasmid and they don't have ampicillin resistant gene. Now we added another antibiotic, and that is tetracycline. Now it's time to find out which of these survival colonies have your foreign DNA. Now what we did, we added tetracycline antibiotic. We added tetracycline antibiotic. Okay, now listen to me very carefully. The non-recombinant, they do not have a foreign DNA, right? Imagine this is your plasmid, this is your ampicillin resistant gene, and this is your tetracycline resistant gene. All right, now in the tetracycline resistant gene, we do not have a foreign DNA. If we don't have a foreign DNA in the tetracycline resistant gene, insertional inactivation will not take place. Hence, tetracycline resistant gene is intact and it is working properly. So if any bacteria which have such kind of tetracycline resistant gene without foreign DNA in it, when you will add tetracycline antibiotic to it, because this gene is working, no insertional inactivation has taken place, this will survive in the presence of antibiotic tetracycline. On the other hand, if I talk about the recombinant one, the recombinant one have the foreign DNA inside of this. The recombinant one have foreign DNA inside of this, because foreign DNA is present here. What is this one? Recombinant one. Foreign DNA is present inside tetracycline resistant gene. This gene has been inactivated by insertional inactivation, and hence this tetracycline resistant gene is disrupted. So when the bacteria which have such kind of plasmid, when it is fed with the tetracycline, that bacteria will die, and this one is not applicable because they are already dead. So imagine only this colony, these two colonies, they survive. So which colonies are these which survive? They are non-recombinant transformants. So from this, what we got to know? We got to know these two colonies, they are transformant. Yes, they do have the plasmid, but the plasmid does not contain the foreign DNA. So they are transformant, non-recombinant. Whereas the first colony to die was this one. This was non-transformant. This is the one which does not have any plasmid at all. And this colony, the last one, is the one that you need to grow further. This is transformant recombinant, because this have your foreign DNA. So now you got to know why we took the replica of the original plate, because the one which dies is the one which we need. So if you will perform the experiment on the original plate, then the recombinant colony will die, and then to whom will you replicate later? There will be no one to replicate. Fine. So by this insertional inactivation, you can get to know which is the one which you require and which you don't require, or which is the transformant and which is the non-transformant, which is the recombinant and which is the non-recombinant. Okay. So NRT means non-recombinant transformant. This is recombinant transformant. This is non-transformants. All right. Okay. So that's about insertional inactivation by using antibiotic resistant gene. Let's talk about further. Let's talk about our another plasmid. Okay, so we are talking about another plasmid now. And what is that plasmid? This is again different kind of plasmid, PUC8. PUC8. P stands for plasmid. UC stands for University of California, where it was engineered. University of California, where it was engineered. And 8 is the index number. Okay. Now what kind of genes it have? First of all, it have this gene which is LAC-Z gene. LAC-Z gene produce beta galacto sidase enzyme, and this have multiple cloning sites. That means just inside of LAC-Z only you will get number of around 9 to 10 restriction site for various restriction enzyme. Okay. Now Here it has another selectable marker, that is ampicillin resistant gene, and then the same ori will be present here. Why? Because without ori it cannot multiply. Moving further, how will we use this bacteria, or sorry, this plasmid, to confirm the presence or absence of your foreign DNA? So for sure, we will add our foreign DNA inside LAGZ gene. We will add our foreign DNA in LAGZ gene by using any of a restriction enzyme which is present in MCS. What is MCS? Multiple cloning site. Multiple cloning site. Okay, now what we do here? First of all, let's understand how this works. For example, antibiotic resistant genes were working by giving the resistance towards a particular antibiotic to the bacteria. Now, how does this one work? See, beta-galactosidase is an enzyme. Okay, so whenever we will add a chromogenic substrate, chromo means that will later on give you the color. This substrate, which is white in color, when it get in contact with beta-galactosidase enzyme, then it will give blue color product. So if my LAGZ gene is intact, it will keep on producing beta-galactosidase enzyme, and if beta-galactosidase is produced, it will convert your white color substrate into blue product. So any colony which is blue in color, that means it has the intact beta-galactosidase gene, and that means no recombinant has been formed. That means no foreign DNA has been added. You don't understand? Let me explain you in more detail. For example, if I have this bacterial plate, now this bacterial plate again have 4 colonies, 1, 2, 3 and 4. Now I need to find out which is transformant and which is non-transformant, which is recombinant and which is non-recombinant. Okay, let's see. First of all, what I will do, let's segregate it. One is transformant, recombinant, that we need. Another is transformant, non-recombinant, and the third one is non-transformant. Okay, so the non-transformant will be the one which have no plasmid. So this can be confirmed with the help of adding ampicillin antibiotic. An ampicillin resistant gene will help us to confirm that which is transformant and which is non-transformant, just the way we have done in the previous one. Now the second type of bacteria is the one which do have the plasmid, and plasmid have intact LAGZ gene. And then we have the third case. It do have the plasmid because it is transformant, and LAGZ gene also have a foreign DNA. Right? These are the three things. If it has intact LAGZ gene, then LAGZ gene will be producing beta-galactosidase. Okay? In this one, because recombinant has been formed, the foreign DNA has been added inside the LAGZ, the insertional inactivation of LAGZ has occurred, so there will be no beta-galactosidase that can be produced. I think now you are clear with this one. Now let's find this out. Now what we will do, first of all, we will add ampicillin in this one. So when we will add ampicillin in this one, all the colonies which will survive are those which are having the plasmid. So first we added ampicillin antibiotic. So these ones survive, it will die because it does not have a plasmid. Now we added substrate. We added white color substrate. So these colonies, they are not producing beta-galactosidase. So if this enzyme is not present, the substrate which is white in color will remain white. So the color of the colony will be white. So if these colonies are white in color, that means these are the one which are transformant recombinant of your choice. Now these colonies, they have beta-galactosidase gene intact. They are producing this enzyme, and white color substrate can be converted into the blue product. So these colonies color will be blue. So when blue color colonies are produced, they are those colonies which do not have the foreign DNA in it. They are transformant non-recombinant. So imagine this colony is blue in color, this one. So this one is transformant, non-recombinant. So this is the second type of technique by which we can identify which one is transformant, which is non-transformant, and the name of the technique is blue-white selection. Blue-white selection. Okay, so these are the white one are the colonies which are transformant recombinant. You will take it out, then you will grow them because you have foreign DNA in it. This plasmid is more better than the PBr322 because in PBr322 you have to change the antibiotics again and again. In this, just in one plate you can do both the experiments. That means you can first add ampicillin to it, and to the same you can add the your substrate. But in this case, whenever you are adding new antibiotic, you have to take the replica again. So after doing this also you have to take the replica again. So you have to take replicas again and again. That's a cumbersome process. So always PUC8 is better than PBr322. Anyways, this was about the plasmid. Let's talk about vectors for cloning in plants and animals. So plasmid was a vector that was used when the host is a bacterium like E. coli. But what if the host organism is a plant and you want to transform a plant cell? Then what can happen under these circumstances, or what vector can we use under such circumstances? So first we'll talk about plants and we'll go for animals. In plants, what scientists saw that there was a bacterium and the name of the bacterium was Agrobacterium tumefaciens. Agrobacterium tumefaciens. So this was a bacterium. It has a plasmid. The name of the plasmid was TI plasmid. TI stands for tumor inducing. TI stands for tumor inducing. Now this plasmid have a piece of DNA known as tDNA. T stands for transfer DNA. So what this bacterium do is this infect dicot plant. This infect dicot plant and create a cancer-like situation in them. So this bacteria, when it infects the plant cells, the dicot plant cells, it will send its TI plasmid to it. And TI plasmid have tDNA, which is a transfer DNA, which will get that can easily incorporated inside the plant cell. Right? So let me just explain you in more detail. Imagine this is a plant cell. This TI plasmid, this is a bacterium. This TI plasmid can easily go inside the plant cell. So that's the property of transformation. So by this property, the scientists were so much glad to say that yes, there is some DNA, there are some bacteria which can naturally do the transformation. They are sending the piece of DNA inside the plant cell, right? But they are sending it for bad purpose. Why? Because when this tDNA will enter inside the plant cell, then it will cause a disease to a plant, and that's a tumor kind of a disease which is known as crown gall disease. So scientists here use some of their brain. They modified this TI plasmid. They modified the TI plasmid, change the tDNA a little, remove all the harmful genes and added some genes like a foreign DNA. Now when the tDNA will go inside the plant cell, it will take the good genes along with that, right? So how did they modify the TI plasmid? So they modified TI plasmid by removing pathogenic genes. Pathogenic means that was causing a disease, and used as vector. Because this bacteria's plasmid can naturally do transformation, and that was used for our purposes. Okay, so this is how this bacterium and this plasmid is used for transformation in the plants. What about animals? So whenever we are talking about animals, we use viruses. Yes, retroviruses. But these retroviruses, they are disarmed. They are disarmed. That means just like TI plasmid, their pathogenic genes have also been removed. Why did we use retroviruses? Retroviruses have a common tendency to infect the human cell just like HIV, but we have removed the bad genes in them. The retrovirus, what it does, it converts your normal cell into cancerous cell. Right? How it will convert your normal cell into cancerous cell? By entering inside your normal cell, just like HIV. So what if we disarm the retrovirus, and then when it will go inside your cell, we will give it the good genes. Take the foreign DNA with it. So when it will be taking the foreign DNA, our purpose is solved. So just like that, we can also use these retroviruses. You understood? So what we did, we removed harmful genes. Harmful DNA from it and added our gene of interest. Our gene of interest. Alright, so this is how retroviruses are used for transforming animal cell, in fact human cells also, because we are also animals. Next we have some viruses that can be used as a vector for bacteria, and these viruses are bacteriophage. What are bacteriophage? Viruses that infect bacteria. So some viruses like M13 and lambdaphage, they are also used as vector. Okay, so do not forget the name. So M13 and lambdaphage, they can also be used as a vector. Now let's solve some question. So the first question on your screen is this. In E. coli, cloning vector PBR322, the restriction site of antibiotic resistant gene tetracycline R. Simply they are asking what restriction sites are present on tetracycline resistant gene. You just have to memorize BAMH1 and SAL1. Answer is 2. Whereas PP11 is present in ampicillin resistant gene. Next. The colonies of recombinant bacteria appear white in color, white in contrast to blue colonies of non-recombinant bacteria, because insertional inactivation of alpha-galactosidase in non-recombinant, insertional inactivation of alpha-galactosidase in recombinant. So they were supposed to write beta, they have written the alpha here, so that's a mistake they have made, but don't worry. Inactivation of glycosidase enzyme in recombinant bacteria, non-recombinant bacteria contain beta-galactosidase. Okay, first of all, the question is asking the colonies which are white in color, they are recombinant, they have already told, but blue one are non-recombinant. Why blue one are non-recombinant? Because in blue one the insertional inactivation has not taken place. Okay, insertional inactivation of beta-galactosidase has taken only in the recombinant one, not in the non-recombinant one. So that's why they are white in color and blue are blue in color. So answer to this question will be 2. Okay, it's quite simple, just use little of your brain. Why white is white, why blue is blue? They are asking that. You have answer here. The white is white because here there is no insertional inactivation. The blue is blue because, here there is insertional inactivation, but blue is blue because here there is no insertional inactivation. Alright, next question is, a gene whose expression helps to identify transformed cell is known as selectable marker. So which gene helps you to identify which is transformant and which is non-transformant? Selectable markers. Next, the given figure in the diagram representation of E. coli vector PBR322, which of these genes can act as selectable marker? Very simple. Out of all these, which are selectable marker? These two. So answer is 2. Next. The given figure in the is the diagrammatic representation of E. coli PBR322. Which one of the following option correctly identify its certain component? Alright. So it is saying that ampicillin and tetracycline are antibiotic resistant gene. True. ORI is origin of replication, not original restriction enzyme. ORI is origin of replication. ROP is the one that produces proteins which is required for replication of plasmid, and hint 3 eco R1, they are not selectable marker. So answer is 1. Next, the 2 antibiotic resistant gene or vector PBR322, very simple, ampicillin and tetracycline. Answer 2, straightforward questions. So I'm not going to explain them, they're very straightforward. Next, a selectable marker is used to. Why? What's the need of a selectable marker? Mark a gene on chromosome for isolation? No. Help in eliminating the non-transformants so that transformants can be regenerated. This is the true, because we want that which are transformant and which are not transformant. We need to get that. Identify the gene for a desirable trait in an alien organism. Select a suitable vector for transformation in a specific crop. Not crop. Here we are talking about the selectable marker. So answer is 2. Next. So that's it about the vectors. I hope you will attend the lecture very nicely and do it well. I'll meet you in the next class. We'll study about the entire processes, how they're done in the biotechnology. For that, stay tuned to the biotechnology chapter with me. Bye-bye. Take care.